Yeah, thank you very much to the organizing committee. So indeed, I'm going to talk about the tool that we developed. And basically, this project started with the observation of Nane Haben, a former PhD student in a group, that discovered that basically when it comes to drug response prediction, so in the clinic or in cell line model, there is a trade-off between robustness and predictive power. So basically, should I wait a bit because I see that everybody's getting in? So I just keep on going. Okay. And so when it comes to predicting drug response, there is usually like 4 main data types that are data modalities that are being measured. At least routinely in bulk, so the mutation, the copy number, the methylation, and the gene expression. And if we go from left to right, we have a gradient of robustness. So basically, if you take 2 replicates, the variability in mutation will be lower than in copy number, etc. It's mostly due to the stability of DNA compared to RNA. But if we look at the predictive ability, the predictive ability of each of the data modalities taken individually, then we actually have an inverse gradient in predictive power. So this was measured by Nane, so published in 2016, where he basically showed that, so here the predictive power is measured as a correlation. So 0 means there's basically no correlation whatsoever, and 1 is perfect. And you can see that mutation is least predictive, followed by copy number. Methylation is a bit more predictive, and gene expression is actually as predictive as all of the other data types taken combined. And so what we want here is to try to find a bit of a sweet spot in this trade-off, and to use gene expression predictive power to guide prediction in DNA-based measurements. And so for this, we turn to an established statistical framework called JIVE, or Joint and Individual Variation Explained. And basically what JIVE does is to take 2 data types that are paired in the samples, so the rows are the same, but the features can be different. For instance, a gene if it's gene expression, SNPs if it's mutation. And then, given these 2 data types, it first computes a joint signal. So basically, two data matrices that are maximally correlated. So all of the correlation of these two data is contained here. An individual signal that's basically orthogonal. So if you look at the correlation between the 2 blue matrices, you don't get anything. And of course, the noise, like in standard dimensionality reduction. But the problem with this JIVE model, or AJIVE is the extension we use here, is that it's based on a Gaussian assumption. So basically, it uses PCA, or principal component analysis, and there are 3 different ways of looking at PCA. So the first one that is very frequently used is the PCA is a linear projector of maximal variance. Usually people also use the fact that PCA is a linear projector of minimal reconstruction error. But what is less used is that PCA is actually minimizing a Gaussian likelihood. So basically, PCA is trying to reconstruct a Gaussian noise model, which in our case is not really faithful to the data, because the data, for instance, in mutation is Bernoulli, or it follows a very different distribution than a Gaussian distribution. So in order to extend the established strategy to non-Gaussian data, we developed Percolate, and for that we made use of an extension of PCA called GLM PCA, or exponential PCA, for people who are more familiar with this notation. And this GLM PCA relies on the notion of exponential family distribution. That's basically a very wide, broad class of distribution, and each distribution within this family is defined by 3 functions. So this eta here, t and a. And this can be seen as modeling choices. So you just define these three functions that are basically based on a priori knowledge you have on the data, and then that will basically assume that the marginal distribution of your data, given a certain parameter, which I'm going to get back to, is proportional to this distribution. Sorry, and which is quite nice because the exponential here can be used in a negative log likelihood with very efficient optimization. And so once we have defined for a single data set, so single data modalities, once we have defined this, the exponential family we want to use, we define the saturated parameters as basically the matrix of the same size as X. So X is the n samples by P genes, or P features in general, P CPG island, whatever. And we define basically the saturated parameter as the single parameter maximum likelihood estimate. So basically for each sample point, we find the theta that maximizes this quantity here. And this here defines basically the negative log likelihood, which we try to minimize. So just minus this quantity here. So now that we have, Importantly, so I'm not showing it here, but this here can be computed very efficiently in linear time. So importantly, what we use now is to basically, instead of projecting the data matrix X, which is usually done in PCA, what we do is we use this saturated parameter to project, basically to find a projection. So usually this GLM PCA is used in a SVD-like approach, so we just find 3 matrices exactly like in SVD. The problem is it's quite difficult then to optimize them together. And to basically scale it to larger data sets, what we do is to use the saturated parameters to compute a loading matrix that corresponds basically to a matrix of basically P, so D latent factors, or D GLMPCs, times the number of features, intercept term, and we try basically to minimize the reconstructed likelihood. So I'm going to walk you through this, this optimization scheme. So we start with a saturated parameter here, we remove the intercept, and then we project with a V matrix on the D principal components. So, and for the people who are more familiar with autoencoders, that's basically corresponds here to the encoder step. And then we just use this, the loadings to map back to the cell view, so to the SNPs or the feature view, and we just add back the intercept term. And so once we have this here, so we can basically try. So once we have this quantity, we try to find the loading matrix here, following a Riemannian manifold constraint, because it's it has to be orthogonal. And we show in the manuscript, and I'm going to walk through the method here, but we show basically that the global minimum obtained with this scheme here is equivalent or better than the one that you obtain with an SVD-like GLMPCA formulation. So, for instance, GLMPCA has been used quite heavily in single-cell genomics. And so once we have this GLMPCA, we can just go to, maybe one thing I forgot to say here is that the good thing is the dependency on the samples is removed. So what we can do with this is basically to plug it into PyTorch, for instance. You just define the 3 functions, and you can use auto-differentiation and stochastic gradient descent to scale it to really large data sets. And so once we have this GLMPCA, we went, so we plug it back to the JIVE model. So we took 2 datasets, XA and XB, that have the same samples, but not necessarily the same features. We define 2 exponential family distribution. This can be very different. So let's say if you have gene expression, you'll take a negative binomial. If you have a mutation, you'll take Bernoulli. And then we find the loading matrices exactly like before. So we just solve 2 problems from before. And using computational tricks, I invite you to look at the manuscript if you want to know more. We compute the score, so basically what is the projection of the samples on this loading matrices. And then to compute basically exactly like in JIVE, to compute the joint model, we stack these 2 matrices, UA and UB, because it corresponds basically to the projection of the samples on these embeddings. We perform an SVD, and the left signal value, so the top vectors are considered as joint signal. And the individual signal is basically the complement of this joint signal here. Of course, the number of joint signal is a parameter to define, and we define it with permutation testing. So for the people who were a bit, who are not really familiar with the mathematical framework, please give me a second chance. So what we do here is the GLMPCA basically can be summed up as follows. So we start with the data sets with n samples and p features. We compute the saturated parameters, and then with our approach that's amenable to stochastic gradient descent, so it scales well, we can compute saturated loadings, so the gene weights, and saturated scores, so sample projections. And once we have this GLMPCA, we plug it into Percolate. That basically takes as input 2 data types with paired samples. We compute the saturated parameters that are here, and then we align the saturated scores to get the joint and the individual signal. So if you like this method and you're curious for more, what I didn't present here is how we do the hyperparameter selection procedure. For this, we use the AIC selection. There is an out-of-sample extension as well. That is the main innovation here, and there's a whole mathematical derivation in the paper. So the first thing we tried is to see whether we can use Percolate to increase the predictive performance of DNA-based predictors, of robust modalities here. And by robust, I mean these 3 data types here. So taken individually, mutation, copy number, or methylation. So each one of them individually is considered robust, as opposed to gene expression. And so the first thing we did was to take one robust modality individually, look at gene expression, and then basically to percolate this robust type into gene expression to get the joint signal, the individual signal, and the noise. And once we have these three restricted signals, we can train predictive models for the 198 drugs that we have. So this here is done all on cell lines. And the first thing we try basically is to compare the predictive power of the joint, the individual, to the predictive power of the whole robust data. So here on the x-axis for each drug, you have the predictive power of the robust data types. Again, it's a correlation ranging from -1 to 1. And on the y-axis, you have the predictive performance of the joint in red and of the individual in blue. And so what I think you can observe here is that the individual signal completely drops, so there's no predictive performance whatsoever in the individual signal. So the signal that is in mutation but not in gene expression is absolutely not predictive. And if you take the signal as correlated with gene expression, is basically yielding a much higher predictive performance. Of course, this is using gene expression here. The copy number, we see pretty much the same thing, and methylation also. So basically, the individual signal doesn't contain anything. Now, of course, we still use gene expression inside the cross-validation loop. So the new samples we have gene expression. So we try to see whether we could generate DNA-based robust type signatures from Percolate in a full cross-validation loop. And for this, we can use the out-of-sample extension that we devised in our paper. So we divide the data in two, in the training fold and a test fold. In the training fold, we did exactly what we did before. So we compute, we basically percolate the robust data type inside the gene expression to get the joint, the individual, and the noise. Now, I showed before that the individual signal doesn't have anything, so it's not really worth it to look at the individual signal here. And from the joint signal, we create a predictive model, again, for the 198 drugs we have in our cell line drug screen, and we apply it on the test fold, for which we don't have any gene expression. We only have a mutation, copy number, or methylation. And so we first looked at mutation, and for mutation, we saw a huge drop in predictive performance. So the gene expression really guides the predictive performance here. And there are basically three kinds of drugs. The first one are the drugs for which we have a big drop on the right here. And these are all targeted agents. So basically, Nutlin-3 is an MDM2 inhibitor, so upstream of P53, and we know that a P53 mutation is really potent and really predictive of response. And the same for PLX472 and a lot of MEK inhibitor here, ARC2, etc., that are basically very, very much predicted by a BRAF mutation or other prediction in a MAPK pathway. So in a way, it's normal that a single feature predicts as well. This one here, for which we have a slight increase in performance, are actually quite interesting because in this case here, we only find chemotherapeutic agents that are known to be for which the mode of action is known to be difficult to predict, for like gemcitabine, for instance. And for this one, we obtain a slight increase in performance, which is quite encouraging. We then went to copy number, for which we see basically the same pattern, and for methylation, for which there's basically no significant differences between the joint and the whole methylation level. And finally, so now I hope I have convinced you that basically the signal the signal predictive of drug response in the DNA-based signal is completely correlated with gene expression, but maybe it goes both ways. And so what we tried basically to answer this question, what we did is to do a deprivation experiment. So we we reversed the experiment I showed before. We took gene expression and mutation because it's the weakest signal, and we percolated gene expression through the robust type. So this yielded the joint and the individual gene expression, and we took this individual gene expression to pass it through copy number. So basically, we only pass in copy number the gene expression signal that is not in mutation. And the signal that is not in copy number either is passed to methylation, and then we basically get a residual gene expression which we can interrogate for predictive power. And that's basically the predictive power we get here. So basically, removing all of the signal that is in mutation, copy number, and methylation is completely depriving gene expression from any predictive power. So to conclude, I showed that presented Percolate, that is a multimodal tool, that is a multi-omic tool that allows the comparison of data sets using GLM-PCA. It's based on the non-Gaussian dimensionality reduction methods. Our implementation of GLM-PCA is scalable and versatile. So here I only show like a few exponential family. But actually, in our implementation, if you have an exotic exponential family you want to use, you can really easily implement it. We show here also that DNA-based signal predictive of drug response is solely contained in gene expression. And reciprocally, we showed that the gene expression signal predictive of drug response is distributed across all of the DNA-based modalities. There's much more in the manuscript, so I invite you to check it out if you want, or to talk to me during the conference or after. And before going to the questions, I'd like to thank the people involved in this project. So my supervisors at the Netherlands Cancer Institute in Delft, Lodewyk, Wessels, Marcel, Reinders, and Marco Loog. Our collaborators in the UMC, Marc van Wiel and Mirrelijn van Nee. There are some positions available, so if you fancy moving to Amsterdam, please contact Lodewyk or reach out to me. Also, for the people who are not really familiar with, who work in single-cell genomics, we have implemented GLM-PCA in SynCE. So if you want to use it for your own method, please reach out to me or look at our GitHub. You can contact me like on Twitter or email, please. Super happy to have any feedback on it. And yeah, thank you very much for your attention.